So recombination, it is the generation of non-parental gene combination. So genetic maps, they are used as a starting point in the sequencing of genome. Y plus and W plus, they are dominant in nature. That is why this plus is being denoted here. Hello everyone, a warm welcome to today's session on the principles of inheritance and variation. I'm Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session, we had dealt about the dihybrid cross. So what Mendel had done in the dihybrid cross. So what is a monohybrid cross? Monohybrid cross is nothing but wherein the cross between two parental pea plants are done, wherein only one trait or character is being considered. That is, Mendel had chosen the height of the plant, wherein he chose one parent plant which was tall and he crossed it with other parent plant which was dwarf. But when it comes to dihybrid cross, here two characters are being considered. So what did Mendel do to do this experiment? He selected two pea plants wherein one parental pea plant had produced round seeds which are yellow in color. So there are two characters here. What is that? The color of the seed and the shape of the seed. And in other parental plant which he chose, it produced seeds which were wrinkled, that is, they were not round, they were wrinkled and they were green in color. And when a cross was done, he found out that it proved his third law, which is called as the law of segregation. So when a cross was done, he found out that it proved the third law, which was coined by Mendel, that is the law of independent assortment, wherein he said that the characters or traits or factors are carried from the parent to the progeny individually. That is, they get expressed, though two different characters are there in the plant, they get expressed separately in the progeny or in the F1 and the F2 generation. So that is what he meant. And to prove his theory, one more scientists, that is three scientists, they came together and they worked on Mendel's law of independent assortment, and they found out that whatever Mendel said during 1860, it was true. But in 1860, since microscopes were not there, people did not believe whatever Mendel said. So after the advent of microscope, the three scientists, they came together and proved that Mendel's theory of independent assortment was correct. To add strength to the theory of Mendel, another scientist, that is Sutton, along with Boveri, they worked on the chromosomal theory and they found out that whatever Mendel had said, it was correct. So 5 scientists, they were the one who proved that whatever Mendel's law of inheritance was proposed, all those were correct. And that is when people from the 1900s, they started to believe whatever Mendel had proposed in his laws of inheritance, that is law of dominance, law of segregation and law of independent assortment. Later on, one more scientist, that is Morgan, he chose the fruit fly, that is Drosophila, in order to study. So the Drosophila, they reproduce very fast, their life cycle is very short, that is about 12 to 14 days, and they give hundreds of progenies, hundreds to thousands of progenies at the time. It is easy to culture them, they are easily available, and the distinguishing characters between the male and the female, it is easy. So because of all these reasons, he chose Drosophila for the experiment in order to further prove the chromosomal theory of inheritance. So what all these scientists proved that was Mendel, he called something as factors, which are nothing but genes that are carried from the parents to the progeny and they get expressed in the progeny. So using these Drosophila or the fruit fly, the scientists, they went in for further experiments. So what they did was, they went in for linkage and recombination. So what is this linkage and recombination? So recombination, it is the generation of non-parental gene combination. Means non-parental gene. it does not come from the parent, but in spite of that, the offspring has inherited the gene. How did the offspring inherit the gene? It is because of crossing over. So you know that during meiosis, what happens? There is reductional division taking place wherein two daughter cells are produced and the chromosomes get distributed to the daughter cells. So during the distribution of the chromosome or during the movement or the separation of the chromosomes in the spindle fiber, there is crossing over taking place. So why these crossing over takes place is, there are two types of arrangement of genes on a chromosome. One is the genes are arranged close together and the other one is the genes are arranged far. So say for example, we consider this to be a chromosome. So I take the example of two genes and I'll call it as gene A and gene B. One more, the same way in other chromosome, gene A, gene B. If two genes are located very close to each other in a chromosome, then these two genes will be carried as such to the daughter cells or to the next generation. But if they are located in different positions far from each other in the chromosomes, then what happens? In one chromosomal arm, one of the gene is located and other chromosomal arm, one of the gene is located. They will not get expressed together in the progeny, but they will get distributed. So this will get separated and this will move separate to another progeny. So that is what happens. Both will not get expressed at a time, or they will not move into, or they will be not carried from the parent to the progeny at a time. So that is nothing but recombination. So in recombination, what happens whenever the crossing over takes place? So for example, if this is the chromosome, so we have the chromosome here, what happens is there is crossing over taking place. So crossing over. So at that time, so if for example, as I told you, A is here, if B is here, again A here, then what happens? This will move to this particular chromosome. So the chromosome will become, so A, B, so it will have A, B and here B. Why? Because this has crossed over with this, or here B. Why? Because this has crossed over with this. So a different set of gene has come to the progeny. So that is nothing but recombination. It is the generation of non-parental gene combination. So you can see here you have the parental chromosome, wherein one is the paternal chromosome and the other one. So this is the paternal chromosome and the other one is the maternal chromosome. So when DNA replication is taking place, what happens? Two different pairs of chromosomes are being produced, wherein one set is from the maternal and the other set is from the paternal, that is the mother and the father respectively. So here what happens is during meiosis, as I told you, if the genes are located, there will be, say for example, the genes are located very far here. So when crossing over takes place, the gene will come here, the B gene will come here. So that time what happens, and whatever gene is present in this chromosome, it will move to the maternal chromosome. So that is how the crossing over will take place. So when crossing over takes place, recombination occurs. That is, a different gene will be transferred to the progeny. So that is what happens in this case. So here also the same thing. So here we have the paternal chromosome, the maternal. When crossing over occurs, you can see that from the maternal, the chromosome has come to the paternal, and from the paternal to the maternal chromosome. So at that time what happens? There will be a pair of gene, there will be a gene or a pair of allele in the daughter cell or in the progeny which doesn't belong to the parent, or it is something dissimilar from the parent because of this crossing over. So that is nothing but recombination. So that is recombination. So recombination is the generation of non-parental gene combination in the resulting offspring. So next there is one more term called as linkage. So in linkage, it is nothing but the physical association of two or more genes on a chromosomes. So here, as I told you, physical association. So these two genes will always remain close to each other and they will get expressed or they will move into the daughter cells together because they are close to each other. So in spite of crossing over, say for example, even though here crossing over takes place, it will be carried as such to the next chromosome together itself. It will not move differently. So that is nothing but linkage. So the physical association of two or more genes on a chromosome is known as linkage, and they do not show independent assortment. That is, they get expressed together at a time. They do not show independent assortment, which is according to the law of independent assortment, what Mendel had said. What he said was, the genes, they get independently expressed in the coming progeny when a part of the chromosomes move into the progeny or during the process of meiosis. So, but what happens here is, because in the case of linkage, what happens, because two genes are located very close to each other, they will be passed on to the next generation as such. So that is nothing but linkage. So I hope you have understood between the recombination and the linkage. So recombination, when two genes are located far from each other on the chromosome, they will move separately or they will move independently to the other daughter chromosomes because of crossing over. But when they're in the case of linkage, what happens? Both the genes are located very close to each other on the chromosomal strand. They will be carried as such without any independent assortment. They will move as such or they will get expressed as such in the developing progeny. So that is nothing but recombination and linkage. So to prove recombination and linkage, we had studied about Morgan earlier. So what Morgan had done was, he used Drosophila in order to study this recombination and linkage, and then to prove the, to prove the chromosomal theory, he used the Drosophila or the fruit fly. While doing so, he found out that there is something called as linkage and recombination. So we had studied previously what linkage and recombination is. So how the genes, they move from, or how the factors or the genes are carried through the hereditary material, that is the chromosome, from the parent to the offspring. That is what he proved, which especially to study the Drosophila, he called them as the sex-linked genes. That is, he used the male and the female Drosophila and he conducted experiments in order to prove the chromosomal theory, in order to especially study the linkage and the recombinations. So what Morgan did was, he carried several dihybrid crosses. Why dihybrid here? I'll tell you, because here also two characters were considered from the fruit fly. That is why. So what two characters were considered? One is the color of the eye. So one Drosophila, that is the male. So previous session, you already know how to distinguish between the male and the female. The easy way to distinguish is the base of the abdomen. So the male will always have a round abdomen, whereas the female, she will always have a pointed abdomen. The abdomen is pointed. So now we know this fruit fly is male and this is the female. So male having yellow colored eye and the female having red colored eye. So what he did was, he made a cross. I told you, he made a dihybrid cross. Why it is a dihybrid cross? To make a cross, two characters are considered here, two separate characters, like how Mendel in a pea plant, he considered two characters, right? Like wherein one parental plant had round seeds, which was yellow in color, and the other parental plant had wrinkled seeds, which was green in color. Two characters. Just like that, in the fruit fly or in the Drosophila melanogaster as well, Morgan, he made a cross between the Drosophila, which had yellow colored body and white eye. White eye and yellow colored body, and also one which had brown body and red eye. So brown body and red eye. So that is what he made crosses between them. That is the yellow bodied, white eyed female and the brown bodied, red eyed male. So next, in one more experiment, he conducted two experiments in order to prove the chromosomal theory related to recombination and linkage. So he made, actually he conducted 2 experiments, wherein in one experiment he made a cross, or he brought 2 Drosophillas and made them mate, wherein one Drosophila, that is the female, it had yellow body and white eye, and the male had brown body and the red eye. So the male is the wild type. So wild types are usually dominant in nature. So wild type. And in one more experiment, what he did was, he chose Drosophillas wherein the female, the eye was white-eyed and it had very small wing. The size of the wing was very small, that is miniature wing. And he crossed it with the male, which had red eye and the wing was very large, that is wild type. So these 2 experiments he conducted. So we shall see what happened when Morgan made a dihybrid cross between the 2 Drosophila flies, wherein the male showed the characteristics of brown body and red eye, and the female showed the characteristics of yellow body and white eye. So we shall, before moving to the experiment, we shall see what are the chromosomes that are involved and what are the genes that are present on the chromosome. So since it is a male, they will have a X chromosome and a Y-linked sex chromosome. So the genes that are present is Y plus and W plus. X will carry the genes and Y is not involved in the expression of any of the genes in this particular case. So Y plus denotes brown body and W plus denotes red eye. So Y plus and W plus, they are dominant in nature. That is why it is plus is being denoted here. When it comes to female, they have the X chromosomes. So they have Y and W, Y and W. And you should see that genes on the chromosomes are located close to each other in both the cases. So here what happens, to what percentage the crossing over will take place, what percentage the progeny will have the exact copy of the genes from the parents, and what percentage recombination we will study. So what happens in this case in the first experiment, we shall see. So Mendel, he brought a cross between the male and the female. So what the male had carried the chromosome X, Y, wherein brown body, which is dominant gene Y plus, and red eye dominant gene W plus. So a crossing is brought with the female. So this is the male with that of the female. So for female, it is X and X. So in the case of female, they have recessive genes responsible for yellow body and white eye. Recessive gene responsible for yellow body and white eye. So these are the parents. So what are the gametes that are involved? The gametes that are involved is X, Y plus and W plus and Y. Then here both are same. So only one gamete, Y and W. So when a crossing is done, what happens? So now a crossing will take place with this, with this, and this also with this one. So now in the F1 generation or in the progeny, what we will expect? F1, Y plus, W plus, it is combining with the chromosome X, Y and W. Again crossing over, we have Y, W with that of Y. So these are the progeny that we get. So what will we get here? So this is this a male or a female? It is a female. Why is it a female? It is carrying both the X chromosomes, that is the sex-linked chromosomes. They are X. So therefore, we know that it is a female. So this is a female and this is the male. So here, what will the character be in the progeny? That is in the Drosophila. So it will have brown body because Y plus is dominant here, and red eye, brown body and red eye. So this is male, it will have yellow body and white eye. So what are the progenies now in the F1 generation after the crossing over? It is brown body, red-eyed female, the progeny, and one more progeny, yellow body, white-eyed female. So this is in the F1 generation. So now again he will cross over in order to see what will happen in the F2 generation. So in the F1 generation, now we have the fruit fly, wherein the female fruit fly is brown body with red eye and the male fruit fly is yellow body with white eye. So again what Morgan did was, in order to again strengthen his research work, he again brought a cross between the F1 generation. Between the F1 generation itself, he brought a cross. So we shall see what happens when a cross is brought between these two F1 generation. What are the gametes involved? We will first see with that. Erase the previous ones that have done. So we shall see what gametes are involved. Y plus, W plus and Y, W. So this is the parental gamete. So consider in the F1 generation one recombination has occurred. So what will be Y plus W, Y, W plus, and next one more gamete, that is Y, W, Y. So these are the gametes involved. So this is parental. Now here recombination has occurred. So this is parental and here recombination has occurred. This is the recombinant. And again here this is also parental. So here one of the gamete in the F1 generation, recombination has occurred. So here why recombination? Can you see here there is exchange of chromosomes. So W plus has moved itself because of crossing over. It has moved to the next chromosome. So that is why here recombinant involved. So when a cross is brought between these, what will happen? We shall see. So I'll erase all these. So what happens when a crossing over is done between the parental chromosome, the recombinant chromosome and the parental chromosome? We shall see. So here this will cross over with this and this one and this one again with this. This gene will cross over with this and this. So we shall see in the F2 generation what is the progenies, how will they look like. So there is Y plus, W plus and Y, W. Then we have Y, W with that of Y, W and then Y plus W, Y and Y, W plus with Y. So we shall see now what are the progenies. So we have, this is the parental, this is also parental. Now here crossing over has occurred. So this is the recombinant recombinants. Both of these are recombinants. Both of these are recombinants. So therefore, in the F2 generation, now we have female fruit fly. There is the progeny is a female having brown body and red eye. So previously we have seen brown body, red eye. Brown body, red eye is represented by Y plus and W plus. So therefore, brown body, red eye. Here again we have yellow body, white eye. Progenies, that is of the F2 generation, wherein they are male. There is brown body, white eye and yellow body, red eye. So these are the different progenies, how they will look in the F2 generation. So the progenies wherein these the females, they have received the same set of gene as that of the parents. So it is retained as such, but the males, they have received the genes that have become recombinants because crossing over has taken place and therefore they have become recombinants, wherein they produced brown-bodied white-eyed male and yellow-bodied red-eyed male. And he found out that from the parental 98.7% and from the recombinant it was just 1.3%. From this particular experiment, he found out that the genes that was transferred from the parent to the progeny as it was around 98.7%, and the genes that got recombined because of crossing over and got transferred from the parent to the progeny was about 1.3%. So what is recombination? The progenies will carry the genes that are dissimilar from the parents, that are not exactly similar to the parents, means their location will be changed. That is what happened here, wherein only 1.3% of the progeny showed recombination, whereas the others, they showed linkage. That is, the genes were carried as such as itself from the parent to the progeny. So this is what Morgan, he found out when he made a cross between the red-eyed male, brown-bodied red-eyed male and the yellow-bodied white-eyed female. So this is what he got. So what he did was, next he went in for another experiment wherein he crossed between the white-eyed miniature winged female and the red-eyed large winged male. So here what he did was, he chose the chromosomes wherein the genes are located not close to each other but from far to each other on the chromosomal arm. So when the genes are located far to each other on the chromosomal arm, how many progenies will get the genes, exactly the same copy of genes from the parent, and how many progenies will have a recombination? We will just see. So what Morgan selected? He selected white-eyed miniature winged female. So he considered first the male. So male, they always have a chromosome, that is X and Y chromosome. So in the X, there is W, which he used for the color of the eye, and M for the wing. So W plus, M plus and Y as it is. And then for female, he used XX, wherein W, Y and W, Y. So W for the white eye and W plus for red eye. So W plus is for red eye. M plus is for large wing, which is seen in the male and they are dominant. And in the case of females, W is for white eye and Y is for miniature wing, that is with small wings. So these are the chromosomes with the respective genes that are involved. So what Morgan did was, he choose a chromosome wherein the location of the genes are far from each other or in different position on the chromosome. So we shall see what Morgan came into a conclusion when he made a cross between the red-eyed, large-winged male and white-eyed, small-winged female. So we will take the parental generation wherein they have the chromosome, that is the male has X and Y chromosome, and the characteristic, what is the dihybrid nature of the male? They have red eye and large wing. So for red eye, which is dominant in male, it is W plus, and the wing, it is M plus. So we shall write it far apart. Why? Because here we are seeing Morgan, he conducted an experiment to find out if the genes are located far apart from each other on different positions on the chromosome, what will happen. So we'll make this changes here, M plus. So this is crossed with the female. So female, they'll have both X chromosomes. So we have again W and M and W and M. So now what are the gametes that are involved? Gametes, we have W plus, M plus, Y and W, M. So now F1 generation, a crossing is done between these. So what will happen? Cross between this. So F1, we will have X, W plus, M plus, and also X, W and M. Here X, W and M, then Y. So this is the F1 generation. So what we have now, in the female, they have the red eye, large wing. Why is it so? Because these genes or alleles, they are dominating over the other. That is why they are dominating red eye and large wing in the female, whereas in the male, it will be white eye and small or miniature wing. White eye, small wing. So again, it is just similar to the first experiment conducted by Mendel. The only difference was the genes on the chromosomes were placed close to each other. Therefore, they were carried as such from the parent to the progeny, but only 1.3% of recombination was seen in the progeny. But in this experiment, what Mendel did, he selected chromosomes wherein the genes are placed far apart from each other. So the gene responsible for the eye color and the gene responsible for the size of the wing, they are located on different positions on the chromosome, that is far apart from each other. So here again, when he brought a cross between these two F1 generation plants. So what happens? So I'll erase these to make space for writing. So what are the gametes involved here? So what Morgan did, he made again a cross between the F1 generation progeny itself. So what are the gametes involved here? X, W plus, M plus, and one more X, W, M. This is one set of gametes that are involved. So these are parental. Next, recombination will occur, like exactly how in the previous experiment it had occurred. The gametes, they will be recombination, that is exchange of chromosomes or crossing over, wherein X, W plus M plus, here we have W and M. This is one set, that is recombinant. And we still have other gametes also involved. Those are X, W, M plus, W plus M. This is also recombinant. One more, X, W plus M. One more, we have the gamete, that is W, M and Y. So this is parent. So this is the parent. So after this, what Morgan did was he made a cross between the white-eyed miniature winged female and the red-eyed large winged male. So these are the different chromosomes that are involved and the genes on that. We shall see when a crossing between the chromosome or when a dihybrid cross occur, what happens. So first we need the parentals. So in the parent, the male, it is having the chromosome X and Y, wherein it has the gene that is responsible for the red eye and the gene that is responsible for the wing size, that is large wing, which is M plus. And here in the second experiment, Morgan, he placed the genes, he considered two characters, that is dihybrid nature, wherein he considered two characters, wherein on the chromosome, the gene that is responsible for the production of the red eye, it is located far from the gene that is responsible for the production or for the trait, that is large wing. So they both the genes are located far apart from each other. So when the crossing over takes place in such a condition, how the resulting progeny will look like, we will just see. So he made a cross between the female X. So here we have W, M, W, M. So this is the male and this is the female. So what are the gametes that are involved? So gametes X, W plus, M plus, Y, W and M. So in the F1 generation, we shall see what happens. So a crossing over will occur. So F1, we have X, W plus, M plus, then X, W, M, and one more X, W, M and Y. So in the F1 generation progenies, the female will have red eye, large wing, and the male will have white eye and small or miniature wing. Small or miniature wing. So this is how it takes place here. So again, Morgan, he was not satisfied with this particular experiment. So again, he made a cross between the F1 generation itself. So he made a cross between the F1 generation. So we shall see what happens. So to make space, I will just erase few things. So here we shall consider the gametes of the F1. So the gametes of the F1, the gametes. So here a crossing is made between the female and the male. So what are the gametes involved? For a crossing to get the F2 generation progeny, we shall see X W plus M plus. So when a crossing over occurs between these two, and then between this and this. So X W plus M plus, then we have X W and M. Before this, we shall see what the gametes are involved. So this is parental, directly from the parental. Next, we have one more wherein in the gamete there is recombination. So X W plus M X W M plus. So this is parental gamete. This is the recombinant. And one more we have gamete involved, that is X W M and Y. So these are the gametes that are involved. So now what happens when a cross is made between these? So we'll just erase this. So we shall see what happens when crossing occurs. So in the F2, we shall see how the progenies will look like. So the crossing of this with this one with this again, and this with this one with this. So now crossing, when it occurs, we shall see in the F2 what will the progenies look like. So we will have X, then W plus and M plus. This crossing has occurred with this particular chromosome. So here X, W and M. So this is parental. As you can see, both are from the parents itself. This is also parent. So both are from the parents itself. The genes have been carried as such to the progeny. But only thing is they are located far apart from each other. Next, X, W, M, X, W, M. Again, this is also parental. We have next set of progenies again, wherein X, W plus M, and then it is with Y. Again one more, we have X, W, M, Y. Both these are recombinants. They have taken from the recombinant. So the genes have been carried from the recombinants. So these are recombinant. Both these are recombinants. So after all these experiments, that is in the second cross or second experiment, what Morgan found out was, so in the progeny, they carried the parental genes exactly 62.8% and 37.2%. So the progeny, and from the recombinant, it was 37.2%. So in the first cross, what Mendel did, he chose the characters wherein the genes were present, both together they were present on the chromosome, wherein only very less set of progenies had recombinant genes, that is those genes which are not present in the parents, or the genes that do not resemble that of the parents. But when he did an other experiment wherein he crossed between two Drosophilas, wherein the genes responsible for the eye color and the gene responsible for the size of the wing, they were placed far apart or in different positions on the chromosome. At that time, the progenies, only 62.8% of the progenies had acquired the same set of genes from the parent, and the rest 37.2% of the progenies had acquired the recombinant genes, or they did not acquire the same set of genes, or there was a variation in the genes that had been acquired from the parent. So this is what he found out. So what it means is that, according to Morgan's experiment, so whenever in linkage wherein the genes are present close to each other in a chromosome, they get expressed as such in the progeny. But when it comes for recombination, when the genes are present in two different, that is far apart from each other in a chromosome, then they will crossing over will occur in the meiosis, and whenever the genes are carried from the parent to the progeny, they show recombination, that is, they show variation in the nature or variation in the gene when compared to that of the parent. So that is what Morgan found. So this was about the chromosomal theory, which supported both Mendel's as well as the other scientists' theory as well. So all these dihybrid crossing over experiment conducted by Morgan on fruit fly also proved Mendel's theories. So that is why Mendel, because of all these experiments, people started believing whatever Mendel had done, and therefore he is called as the father of genetics. So there is one more thing. So all sorts of chromosomal studies, everything, the study of the cell, the nucleus, inside the nucleus, that chromosome that is present, location of the genes, all that were improving as the years passed by since the 1900s, because of the invention of the microscope, or in other words we can say because of the improvements in the technology of the microscope. So that is when gene mapping came into existence. So what is this gene mapping? So far we had studied the position of the genes on the chromosome. So we studied about the linkage and the recombination. So in linkage, the genes are located close to each other, whereas in recombination, there is crossing over occurring when the genes are located far from each other on the, or very far apart from each other on the chromosome. So to study all that, to make it more easy for the people to understand, or for the other scientists who are experimenting on genetics to understand, Alfred Sturtevant, he used the recombination frequency between gene pairs, and wherein he measured the distance. So if these are the chromosomes and these are the different genes that are present on the chromosome, he started to measure the distance between the genes. So exactly in which place of the chromosome the genes are located. Why? Because chromosome is nothing but it carries the genetic material, that is the DNA. So in the chromosome we have the genetic material from, or that is the DNA. So the genes are located on the DNA. So exactly where, in which position they are located. So distance between these two genes. So all that he started measuring, and that measurement came in the form of a map. It is called as gene mapping. So between the genes, and he mapped their position on the chromosome, exactly where there is located, where it is located on the chromosome. So genetic maps, they are used as the starting point in the sequencing of genome, and therefore that particular genetic mapping, all that was called as the Human Genome Project. So genetic maps, they were used as a starting point in the sequencing of the genome. So sequencing of genome is nothing but in the chromosome wherein the DNA is present. So exactly in which location or which position the genes are there, that those will be mapped. So you can see here, this is a chromosome wherein two arms of the chromosome you can see, and here the genes are located exactly in which place. So whether they are located close to each other or whether they are located far from each other. So all these mapping is done. So all this is computer based technology. So that is that credit, it actually goes to Alfred Sturtevant, wherein he used the recombination frequency to measure What is the distance between two genes on a chromosome? How far they are located? What position they are located on the chromosome? So all these mapping actually helps people to find out the genetic disorders or any mutation occurring in the gene or on the DNA that can be easily found out because of genetic mapping. So genetic maps, they are usually used as a starting point for sequencing of genomes, and it was done under the name of Human Genome Project. So nowadays, all the genes, especially the genes of Drosophila, the rats, then we have Arabidopsis thaliana, which is a plant, rice plant, wheat, all the genomes of almost most of the plants and animals as well as the microorganisms, they have been mapped and stored in the databases in the computer. So this is about genetic mapping. So in today's session, we learnt especially about linkage and recombination, and in order to prove what happens when there is linkage and recombination, and in order to prove the chromosomal theory, we studied about the experiments, that is 2 important experiments conducted by Morgan on the fruit flies, that is the female fruit fly and the male fruit fly. When crossing over is done, what happens, and also the different locations of the genes on the chromosome that is mapped. The process is called as gene mapping. We have studied about that. So in the next coming sessions, we shall study the polygenic inheritance. That is when more than 2 genes are there, how they get inherited in the progeny. Polygenic inheritance we shall study, and we shall also study about the sex determination in human beings and human beings and honeybee. So we shall also study the sex determination in human beings and honeybee, the methodology followed behind the sex determination. So I hope you have understood this session very well. We shall meet in the next coming sessions. Thank you.